Thank you very much for a very generous introduction. I'm truly honored to be here today, and thanks to Dab and his team for organizing a wonderful event. It's a great hospitality here. This is not my first time in Pakistan, but I feel very special today. Thank you very much. So I've been asked to talk about how we can use novel NDV vaccine as a candidate vaccine for NDV, but also against other pathogens within the poultry. But before I go in there, I just wanted to take a moment to introduce what is Lancaster University and Lancaster itself. So Lancaster is really up into the north of England, if I may use the cursor here. So it is very close to Scotland. The only town in between Lancaster and Scotland is Carlisle, and we are at the Morecambe Bay. So Morecambe Bay is one of the most beautiful bay which connects the Irish Sea to the Isle of Man and to the Ireland. So it's around 1.5 hour drive from Manchester, Nottingham, and Liverpool. So not only that, it's a beautiful place, but we are very small in town. I think only 150,000 people living there, very beautiful beaches. And we are having called Lakes District, which is the national park for the northwest of England, a very beautiful place to raise children. So not only about its beauty, but we stand top 10 in the country, which means the Lancaster University, according to all three criteria used to classify universities, Lancaster University stands in top 10, which is a really great thing because, UK is a country of university where it's Oxford, Cambridge, Imperial, UCL, and standing top 10 among those universities is incredibly competitive. So within the one of the faculty of the four faculties in the university, we have a faculty of health and medicine. Within the faculty of health and medicine, we have a group called infectious diseases transmission and biology, and that is where my group works. So my group consists of around 18 people at the moment. Some of them have left and some are in the process of transit, and some of them has been quite deeply involved into different work. And I put one QR code here to go into more detail about what kind of work we are doing here, and on the website that is detail about the overall activities that we are doing at Lancaster University. And I think one of the things that we are very proud is that we have been at the forefront to break down hardcore science to the general public to really convey the message that has been very hard to digest otherwise. So my group in the Lancaster University has been categorically funded by the BBSRC, which is one of the government funding body like HSC and Newton Fund, British Council and Royal Society, and some of the, of course, industry and the government organizations. But recently, I'm very honored to have a couple of very good grants from Innovate UK and Innovate UK Business Growth, as well as International Science Foundation Fund to really develop novel vaccine and advanced diagnostics. So really jumping into the topic, that is what I was supposed to say. I genuinely wanted to apologize. The funny font that you can see over there, that is an incompatibility between Mac and Windows. So please spare me for that. So what I really wanted to go very quickly going to what are the challenges in controlling Newcastle disease virus? What is the genotype distribution? Can we really make it thermostable, which means that it can survive under a very high temperature? And can we use this whole Newcastle vector vaccines to apply in the field conditions? So I don't want to take you into the introduction and the background of these because these audience are already very well prepared, and Chun Chun has already given the background. What is the Newcastle disease virus? But we do know that infectious diseases remain one of the most problematic, most economical burden onto the productivity and the health and well-being of the poultry. And my topic has always been and will remain around the Newcastle disease virus. So no introduction and the background about this one. All I want to say is that it is very lethal. And with the allogenic Newcastle disease virus, almost all birds, those are infected, could die, would have a significant bearing and impact on to the trade and the profitability of the farmers. So you might have been thinking, what is happening really with the Newcastle disease virus, and it is still endemic even 100 years after its first identification. And well, there are many factors, and some of the factors are very generic and common for literally every viral infection we talk about. And I'm not going to go into all of these, but all I want you to do is the last 3 points, those are colored red in here. So one thing is that we need to make a vaccine, those are country specific, those are region specific, because we can't make a vaccine that fits for all. And the second thing is that we have to match the antigen. As Munir Iqbal mentioned, that is not the genetic antigenicity, but also is at the epitope mapping and so on. And thirdly is about the thermal stability of the vaccine, because you can imagine the temperature we are living in that situation, the disease could flourish without having effective thermostable vaccine deployment. So all of these need to be considered to have a better and improved vaccine. So when I talk about the genetic diversity of the NDV, there are only one serotype. What does that mean is that literally all Newcastle disease viruses you put into the chicken and you take the sera, every Newcastle strain can be neutralized. That means it's a single serotype. But that doesn't mean that it is single genotype. There are plenty of genotypes out there. And now more and more research is coming up that majority of those genotypes are very diverse antigenically and genetically. So when I began working on the Newcastle disease virus during my PhD back in 2012, we did the complete genome sequence, and we really wanted to nail down what is happening in the world of the Newcastle disease virus. That was the time when new and new lineages, new and new genotypes were being introduced into the field. For example, Giovanni Catoli from Italy, they have introduced a new genotype called genotype 7 that is mentioned over here. But when we did the complete genome sequence from Pakistani isolates, we say that no, there is no separate genotype because these blue color sequences are from Pakistan. So literally all this represents is a single lineage, and that lineage is lineage 5. So what we did, and we moved on and we defined a criteria how you can classify a genotype and the lineage and the sub-lineage and the genotype. So simple is that if you have a similarity more than 80%, those will be called a sublineage. If you have a similarity of around 80%, then you call it a lineage. And less than that would be class 1 or class 2. Thereafter, we took a countrywide approach, literally from every district. We have collected over 500 samples and we sequenced every possible Newcastle disease outbreak in a couple of years. And we mapped about the diversity of the Newcastle disease virus in Pakistan. Of course, as you can anticipate, genotype 7 was the most predominant one, but in the KPK, we also found genotype 4 as well. So that does indicate that the diversity of the Newcastle disease virus in the country span not just the genotype 7, but across other genotypes as well. So what happened really, since we began doing these studies, people were coming up with a lot of lineages, a lot of genotypes, and they keep saying, oh, genotype 7, A, B, C, D, E, F. But is this really a realistic approach? I said, no, it wasn't, because it created a lot of confusion. Because, for example, you can imagine if isolate is classified as genotype 7B, and if you are going to use a genotype 7A for the challenge study, how are you going to correlate all of those immune protection and the cross-matching off of that? So to address this problem, we began building a consortium. Everybody in the planet who were working on Newcastle disease virus, we come up with a plan to introduce a new nomenclature for the Newcastle disease virus, the same way as it is being used for the influenza. For example, instead of calling genotype 7 A, B, C, and D, you call genotype 7.1, 7.2, and 0.1.2. Similarly, as the evolution goes, you can carry on reclassifying them as a more systematic approach to have the same system that literally every country and every group who is working on Newcastle virus could use that. So how you can use these resources, if you go onto this publication, you can download a data set that is already available free, and you can include your sequences in that data set and do the phylogenetic tree. You will exactly know where your sequences is belonging to. And I take this forum to introduce this one such that everybody working on Newcastle disease virus should start using this new and revised classification system. So if we really look on to what kind of genotypes are circulating around the globe, there are plenty of them, but I really wanted you to focus on this blue colored one, because that is where is really the one that is creating problem, not only in the Southeast Asia, but also in the Middle East, and especially talking about Pakistan in the context of this one. But of course, you do know that genotype 13 has been isolated by Khaja Saab and his team as well. So this genotype 13 is also emerging concern globally as well. So all what we wanted to say so far is that genotype 7, regardless of its subgenotype, is really the main concern that we have to tackle moving forward for global eradication and control of this Newcastle disease virus. And one way to do that is, of course, you can look into what kind of vaccines are currently being used. There are two types of vaccines. One is the live and another one is the killed. And there are different approaches those are being used. But the key message that I wanted to pass in this slide is that you can imagine in the blue arrow at the bottom, this is the vaccine strain. and the actual virus that is causing the disease is at the top of the tree. So you can imagine that literally it's a big genetic difference. So you have to compare apple with apple, otherwise you won't be able to provide protection that is required to protect the bird against these field strains. So genetically, they are highly diverse, and similarly, they are diverse genetically too. So is the genotype matching is the realistic strategy, and we should definitely think about this one. And before going into this one, I wanted to give you a little bit of idea how this whole vaccine thing works. Is because when virus infects a host, it infects a special cell. The cell is taken by the antigen presenting cell, and as a result of that, you have a lot of antibodies circulating into the blood of the animal. So if you take the blood, you separate the sera and look for the antibody. Those are the antibodies that you really have to think about when you talk about the vaccine. So when we talk about the antibodies, those are actually Y-shaped small molecules. These are proteins that bind and latch onto the virus and really neutralize them. So for influenza, for example, you need 70 antibodies to neutralize one virus. Same is the case for around 200 for the rabies and the polio, around 30 antibodies. But how much is really needed for the Newcastle disease virus, we don't really know. And what we can do is that we can blindly work onto those antigens. Those are critical for the design of the vaccine. And those are F protein, which is the fusion, and HN, which are the hemagglutinin neuraminidase protein. So from this point onward, I will be only using F and HN. So if we look onto the diversity of the F and HN, what is F? F is this head-shaped protein that is lying on the surface of the virion as like a crown. If you look a little bit more into the three-dimensional structure of this, like how exactly it looks like, you will be able to map out that there are some sites onto the head of this protein that is critical for inducing the immunity that is required for protection. But on this blue color alignment, you can see that based on different sequences available into the public domain, there are a lot of variation out there. So you have to consider these variations to design best and compatible vaccines. And how all this works is that you have a vaccine strain you given to the bird. Bird has a lot of antibodies, and those antibodies are against N and F proteins, and those are shown here with the circle, triangle, and into the rectangle. So what happens that these are those antibodies that bind onto the virus when the field strain comes in and neutralize this virus, and that is how the vaccines provide the protection. Right? But what happens is that if the virulent strain, the one that is causing the disease, have the epitopes that are different than the vaccine strain, what you will end up is that only few of those antibodies will be valuable. The rest of all antibodies are useless because they are not binding to those epitopes. Those are critical for protection. So those will be escape antibodies. Those are the antibodies. Those are spared. So how you can really counteract this problem? There are only two ways to do that. Number one is that you raise a lot of antibodies against the vaccine strain, which is Lasota or anything else, so that you literally have a gazillion of antibodies in the blood, and every antibody will bind to the epitope and neutralize the virus. The other approach is that you will then use a field strain that is very closely genetically and antigenically related to this one, so that every antibody raised is valuable. So there is no free epitope, there is no free antibodies, and every antibodies is critical for the vaccine efficacy. Right? So other than that, there is no other approach. So how people have been trying to do this approach, that they've been converting a lantogenic strain, which is an apathogenic version of the NDV, into something that is very safe and effective, but that approach hasn't been really working very well. The other approach they have been doing is that they were mutating some of the amino acid within the NDV protein to convert pathogenic into non-pathogenic, and then use that one for the vaccine approach using reverse genetic technology, but those are not working as well. So what we came up with a plan is called codon de-optimization. So I don't want to take you in a hardcore science here, but all I wanted to mention is that the ribosome identifies some of the nucleotides onto the messenger RNA and then translate that one into protein. But some of the messenger RNA codes are more favored by the ribosome than other codes. So what we did, we introduced those codes into the messenger RNA. Those were not favored by the ribosome. So ultimately what we have is a vaccine that is attenuated because some of the proteins will be expressed less compared to the other proteins which are not codon de-optimized. So as a result, what will happen is that if you are going to use a lantogenic strain of the NDV, which is a non-pathogenic version of the NDV, you will be having a very localized immunity only in the upper respiratory tract of the bird, which will not be enough to provide protection against these field strain at the system level. But what you would do is that if you use the valogenic but non-disease causing vaccine strain, you are going to introduce the immunity throughout the body of the chicken. So you can imagine you are only telling the upper respiratory tract to raise the antibodies, that's going to be limited. But if you're going to tell the whole body that raise the antibodies, that's going to be a lot more protective. And that is what we have achieved using this novel vector. So leaving here, Newcastle disease virus is a disease of tropical and hot countries. And as you know, I think you have witnessed at the coffee break that it was really hot out there, wasn't it? So you can imagine the virus would be able to survive because it is infecting the host inside the chicken, but the vaccine, it is not in the host, it requires protection. And we do know that the summer temperature is a very hazardous for the vaccine safety and its stability. And around 50% of the vaccine is literally wasted because of the thermal liability, the thermal sensitivity of the vaccine. And we sometimes don't really consider that part is because it doesn't matter whether it is degraded or not. We simply use that vaccine into the poultry flock without realizing that actually that is useless. What we need to do is to really protect this thermal stability. And main problem with really all of this phenomenon is that it is irreversible. Meaning that if the thermostable vaccine is not really considered at the final stage of the application, is not going to really achieve all that what you needed. So we just want to take 1 minute to explain how this thermostability and thermoliability works. So you would have a bird, you have given a vaccine on the upper respiratory tract, it binds to some of the cells, those are in the upper respiratory tract that are pneumocyte, some at goblet cell, and start producing antibodies. And those antibodies are the ones that are required for vaccine-induced immunity. And then what you happen to see is that if you have a virus that is thermostable, meaning that it does not respect the increasing temperature, you have a very beautiful decoration of these proteins H and F onto the surface. And if you take the electron microscopic images, you can see a very beautiful serration of these different H and F protein onto the surface of the virion. So this is the shape of the virus that is thermostable, so it does not respect any increasing temperature. But what happens if you give a temperature to the virus, all these proteins, those are on the surface of the virion, start peeling off. They just start fluffing. Because of this peeling off effect, the virus will not be able to infect, and that is how the virus will not be able to induce the immunity. And what does that mean is that if you take the electron microscope image of this thermolabile virus, it does not have those H and F protein onto the surface, meaning this virus will not be able to infect and induce the immunity. So what we really did, we took an approach is called a negative surface charge. We introduced those nucleotides inside the HN protein of the Newcastle disease virus that increase the negative charge onto the amino acid. I'm trying to make it as simple as possible. So on the right side is the more reddish color of the HN protein, which is thermostable because we introduce more negatively charged residues. On the left side, you have thermolabile, which has less number of negatively charged residues, more of positively charged. So then you use the reverse kinetic system to rescue these viruses to use for the application in the field. So we have done a lot of work on there, but I just wanted to show you one figure is that if you increase these two, so if you take two tubes, in one tube you have a Lasota vaccine, and in another tube you have this thermostable vaccine, and you heat it up at 56 Celsius only for 10 minutes, you will have significant, at least 10 log reduction into the Lasota infectivity. So you can imagine if one vaccine has reached to 56 Celsius in one way, shape, or form, it would have lost almost 50% of its infectivity. So taking this matter forward, we developed a genotype matched vaccine that provide systematic immunity and is thermostable. So using that system, we filed a patent, and that patent has already been protected around the globe. So literally nobody can use the Newcastle disease with those features that we introduced anywhere on the planet. So all those features are attributed to the Newcastle disease for its ability to use as a vector for immunization in the poultry sector. I don't want to go into each of those, but all those are the key feature that make a vaccine one of the best model vaccine for poultry diseases. So using all of that system, what we did, we have produced half a dozen of these vaccines against almost every disease of the poultry, and more on their way because we are introducing some of the mutations in there. So just 1 more minute to go through that. This is the challenge study where we vaccinated the birds with this novel vaccine vector, and then we challenged with the genotype 7, and all the birds, those were vaccinated, survived compared to the control one. Then we took the IBV approach, and IBV is very unique because the S protein of IBV is very big, it's around 4 kb, and stabilizing the IBV into the NDV is a quite a nightmare. But because of this novel genetic modification within that, it has accommodated quite well until 20 passage at least. So as you can see that not only the birds, those were protected, but into the Alice spot, you have a high level of antibodies. Lastly, I wanted to mention the vaccine that we developed for H5. What we did that instead of taking only the hemagglutinin protein, we also took the NP and the 3 M2 genes. So M2 genes are really important. The proteins are important for the proton exchange. NP are critical for T cell immunity. HA is for the humoral immunity. We took all of these 3 into the backbone of NDV and stabilized that and used that one as a vaccine candidate. So lastly, all I wanted to say is that a genotype match approach is really critical if we wanted to control the infection, not only in Pakistan, but also any disease endemic country. Subgenotypes are really something that we don't need to worry at all, because those are only for the genetic and epidemiological studies, not for the vaccine purposes. We need definitely improved vaccine. Thermostability is one of the things that we need to pay attention to. And using our novel vector system, which is allogenic, not because of its pathogenicity, but only because it goes in systemic and it's attenuated. It does not cause the disease, provide a superior system level vaccine. That is what is required in the field application. With that, thank you very much, everybody, for your attention.